Good day, Professor Reed. How did you start your work in research?
Well, my work in genetics research started in a very strange way because actually I do not have any training in genetics or indeed in any branch of biology. I actually trained as an organic chemist, and in the British system you specialise very early in school, so I did chemistry, physics and maths in school. At Cambridge University I did chemistry and biochemistry. For my PhD I worked on chemistry, but it was nucleic acid chemistry, it was RNA chemistry. So I studied RNA, but as a chemist, not as a biologist or a geneticist. But eventually, when I worked in Manchester University, I started going to the seminars in the Department of Medical Genetics because I thought these are the most interesting seminars I've ever been to. I thought chemistry is a bit boring really, and the genetics is so exciting, so I started going to the seminars in medical genetics, and eventually the kind professor gave me a job in the medical genetics department. So that was how I started research, and that was back in 1977. Now in those days you couldn't do anything useful with DNA in medical genetics. There was nothing you could do that was relevant to patient care. So initially I worked mainly on introducing computer databases into the department. In those days you had to write your own database programs, so I used to write Fortran programs and so on. And also I worked on neural tube defects. I was one of the group that showed that if you supplement pregnant women with folic acid, then that reduces the incidence of neural tube defects. But then in about 1980, there were two important developments which showed that DNA will soon be important in clinical genetics. There was a paper from Botstein and colleagues in the American Journal of Human Genetics, and that suggested that you could use DNA polymorphisms as tools to map disease genes in the human genome. And in addition, there was a paper from one of the obstetricians in California showing that you could use DNA polymorphisms to track the sickle cell disease gene in patients and also to discover carriers for sickle cell and do prenatal diagnosis. So from about 1980, I thought it's going to be very soon a good thing that I know something about nucleic acids. But the techniques which I had used in my PhD, which was back in 1964, these were all completely old. And so I went to Leiden in the Netherlands, and there I learnt the newer methods of extracting DNA, of digesting it with restriction enzymes, of running agarose gels, of preparing radio-labeled probes, doing southern blots, and so on. So I came back to Manchester in 1981 and I set up our DNA corner in the cytogenetics laboratory, because in those days the cytogenetics laboratory was our only laboratory, and with one of the cytogeneticists we worked on bringing those techniques to Manchester, and quite soon the first linked polymorphisms became available, so we could start trying to track the genes for Huntington disease, for Duchenne dystrophy, for polycystic kidney disease. Those were the three main groups where we had registers of patients and families, and we worked trying to track those through the families and to be able to offer carrier testing or pre-symptomatic diagnosis. And that gradually started working. At the same time, I was actually employed by the university, not by the health service, and so really, as a university person, I should not have been running a diagnostic laboratory. I should have been doing research as well as teaching and so on. And so I started research. I was very interested in the idea of using DNA polymorphisms for genetic mapping, but in those days it would have been quite impossible to map an autosomal condition, because if you don't know where your disease gene is anywhere in the genome, you need about 300 or 400 polymorphisms in order to cover the whole genome. And each polymorphism, you have to do southern blots. It's a very difficult technique. It takes perhaps 2 weeks to get good results. So you can't study 300, 400 polymorphisms with those techniques. So I started by looking at an X-linked condition, because we had a good bone metabolism unit in Manchester, and they had quite a lot of families of people with X-linked hypophosphatemia. So the clinicians could show me the pedigrees, they could say who was affected, who was not affected, they could get DNA samples for me, and so we could do linkage analysis. And if you know the condition is on the X chromosome, you only need maybe 10 or 15 probes in order to map it on the X chromosome. So we did that, we were able to show where the gene mapped on the X chromosome, and eventually, in collaboration with researchers in London and in Canada, we were able to identify the gene. So that's how it all started.
And could you compare that time with the time we are living now, and how genetics changed?
Oh, it is so different. It really is so different. When I look at what we could do in those early days, it was so difficult to find out anything. So the accent then was really on trying to discover a variant. It was not easy to find variants, and so you tried to think of a clever idea why your disease might perhaps map to chromosome 2, for example. I worked later on Waardenburg syndrome, autosomal dominant condition with hearing loss and rather strange pigmentation. So a typical Waardenburg patient has one blue eye and one brown eye, and they have a white forelock in their hair. They're not all like that, but that's a typical thing. And for various reasons, I was very interested in Waardenburg syndrome. And in those days, you tried hard to think, well, where might the Waardenburg gene be so that I can concentrate my probes on that area of the genome? And there was some evidence from one patient in Japan who had a chromosomal abnormality, and from some mouse mutants which had rather similar phenotype, and we thought this maps perhaps to the bottom end of chromosome 2, so that was where we concentrated. We were lucky, we found it. So in those days, the really important thing was to find a variant. Well, nowadays, when you run your exome or genome sequencing, it immediately tells you for the exome 20,000 variants, for the genome 4 million variants. And so now the question is not how can I find a variant, but how can I sort through this enormous long list of variants to try to discover the one which is the important variant. So it has changed a lot. And in particular, I think, it's changed from a phenotype-driven approach to a genotype-driven approach. And what I mean by that is, when I used to work on Waardenburg syndrome, so I would ask the clinicians with whom I worked to diagnose the patients for me, and I collected patients who had the label Waardenburg syndrome, and then I looked at the relevant gene. It was called PAX3. I looked at the PAX3 gene in these patients, and so in the end I had a list of the sort of mutations in the PAX3 gene which you would find in people with Waardenburg syndrome. And that was a phenotype-driven approach, because I started with the clinical phenotype and I tried to look at the possible genotypes. Well, now you do it the other way around, because you first of all do your exome, you find a huge load of variants, so you start with the genotypes, and you can then look in your data at the PAX3 gene, and you can say, well, what variants in the PAX3 gene do I find in patients with Waardenburg, in normal controls from the EXAC or NOMAD databases, and maybe in people with completely different clinical phenotypes. So what we see now is that many variants which we thought were strongly pathogenic it turns out actually they're not very strongly pathogenic. They have a reduced penetrance, and we also find that often variants in a gene cause not merely the syndrome that you were interested in, but also other phenotypes. So we have a very different view now of genotype-phenotype correlations, and it's all becoming extremely interesting.
Yeah, so it changed a lot, but the amount of work, it's getting even greater, I think.
Oh yes, it's wonderful, yes.
Could you tell us what was the critical moment in your career?
Well,
or at least one.
I mean, in some ways the critical moment was when the kind professor in the medical genetics department said to this chemist, come and work in my department. But in terms of science, I think the critical moment was when we were able to discover the gene which was mutated in Waardenburg syndrome, because it turned out, we were very lucky, it turned out that this was a very, very interesting gene, because the reason why I was interested in Waardenburg syndrome, well, there were several reasons. I mean, first of all, it was possible because we had a very good audiologist who had collected a lot of families who made really very clear diagnoses, and that is absolutely critical. Without that collaboration, nothing would have worked. So it was possible, we had the material. But the other thing is, the Waardenburg gene turned out to be very interesting, because if you ask, what goes wrong in somebody who has hearing loss and this funny pigmentation? And the answer is it's to do with melanocytes. And in the early embryo, melanocytes start off in the neural crest and they migrate through the embryo. And what's happened in Waardenburg syndrome is these melanocytes have lost their way. They've failed to arrive at the correct place. You need melanocytes in your inner ear for correct hearing, and you also need it for the pigmentation. So I thought, this is one of the big questions in biology. I mean, you start off as a single fertilized egg cell, and you grow into lots and lots of cells, but how do these cells end up forming the shape of a person with legs at the bottom and a head at the top? How do they pile up? Somehow, they must know where they are. And I thought, maybe the gene which goes wrong in Waardenburg syndrome, when that gene is working, perhaps it's telling melanocytes where they are. So I thought this condition, which is not very important clinically, I thought biologically it might be very, very interesting. And we were indeed very lucky, because when we identified the gene, it turned out that this was the first example of a human inherited mutation in a class of genes which people had been very interested in, and they had been looking for mutations but never found them, so-called homeobox genes. And homeobox genes were identified from some very, very strange mutations in Drosophila fruit flies, in which the fly appeared to have been built in the wrong way. So, for example, one of the Drosophila mutants, on its head it should have antennae, but it had legs. So it had been built in the wrong way. And there were a series of these mutations. They were called homeotic mutations. And in Drosophila you have the techniques which can identify the genes much more easily than in humans. And so it was known that all the genes which produced these strange homeotic mutations all encoded proteins which contained a sequence of 60 amino acids called the homeobox. Later it was discovered that humans also had homeobox genes, very similar to the Drosophila ones. They're extremely strongly conserved in evolution. And so, of course, then people asked, well, what happens in humans if a homeobox gene goes wrong? Well, we don't end up with legs on our head, but what does happen? And so people were really interested to try to find human inherited homeobox gene mutations. Well, the first one was our PAX3 mutation, so that was a turning point in my career, because having found a really interesting gene, then I got invited to all the nice meetings in sunny places to go and talk about it. So that was good. I was very lucky. I really was.